Welcome everyone. Here we'll move on to with our lesson, that is biomolecules we are studying. In the previous video we have seen the structure of proteins. We have seen that proteins are found in 4 structures, that is it can be found in primary structure, secondary structure, it can be found in tertiary structure, and it can be found in quaternary structure. So proteins can be present in quaternary structure, that is it can give us 4-dimensional view. Now coming to this another topic, we have finished with all those micro and macromolecules. In micromolecules we have seen amino acids, water amino acids, then we have seen monosaccharides, we have even seen nucleotides, nucleosides, all those things we have seen in biomolecules. And in that is macro biomolecules, that is bigger molecules, we have seen proteins, we have seen we have seen proteins, we have seen polysaccharides, we have seen nucleic acids, we have also seen the structure of lipids. So now we are finished with all those biomolecules. We have studied almost all those bio micromolecules as well as bio macromolecules. So now coming to this another topic, here it is nature of bond linking monomers in polymers. We have said that polymers, that is protein, it is a polymer. Protein, it is a polymer. This polysaccharide, it is a polymer. Nucleic acid, it is a polymer. How are these polymers formed? How are these polymers formed? So these polymers are formed when small monomer units join together. When small monomer units join together, then only a large polymer is formed. When this small units, you just take, this is a small unit, this is small unit, this is small unit, this is small unit. When these small units, that is monomer, the small units is called as monomer. Okay, this one monomer, another monomer, like this many monomers join together, we form a polymer. Poly means many, mono means one unit is small unit is joining together to make a big molecule, that is a polymer. So we said that monomers combine to form polymers. So how are they combining? How are they combining? What is the nature of bonding? Because they are combining with the bonds, with the help of the formation of bonds. These two monomers are combining with the formation of bonds. These two are combining with the formation of bonds. So what type of bond? By what type of bond? These monomers are combining and making into, that is changing into a polymer. So that is what we will be seeing in this video. So what is the nature of bonds? Proteins, they are joined together by amino acids. So these amino acids binds together and form proteins. So here now which bond is present in between those amino acids? How are those amino acids binded together so that a big molecule, that is protein, can be formed? We already know that proteins combine, that is proteins are nothing but it is the that is amino acids joined together by peptide bonds. Amino acids joined together by peptide bonds. We have also seen this nucleoside combines with sugar. We have seen how not nucleoside, how a nitrogenous base combines with sugar and forms a nucleoside. We have even seen how a nitrogenous base combines with sugar as well as phosphate to form a nucleotide. So how are these bonds formed? How are these bonds formed? So what happens when nitrogenous base combines with sugar? So the bond which is formed is glycosidic bond. And when the phosphate is added to this sugar, so the bond which is formed is ester bond. It is ester bond. Phosphodiester bonds are there. So let us see what type of bonds will be bonding the different polymers. So here we have already seen proteins, or we can, that is proteins are also called as polypeptides. Proteins are polypeptides are joined together by a bond called as peptide bond. Joined together by a bond called as peptide bond. Let us see how it is, how peptide bond is formed. So here this is one amino acid. Amino acid, that is, it consists of an alpha carbon. To alpha carbon 4 groups are attached. One is the carboxyl group, one is the amino group, one is the hydrogen group, and one is the R group. So this is one amino acid. Now one amino acid is joining with another amino acid. This is another amino acid. So I'll remove this first. Okay, this is one amino acid, this is one amino acid. When these two join, then only a long chain, that is, a chain will be formed. So how these two will be joining? This is one amino acid, this is one amino acid. So what happens is, what happens when they join? How they join? So when they join now, here OH from this carboxyl group and a H from this amino group. OH from this carboxyl group and an H, one hydrogen from amino group and one OH from carboxylic group is removed in the form of water molecule. So here what is happening? Here oxygen is coming, hydrogen is coming out. Here one hydrogen is coming out. So how many hydrogens are there? 2 hydrogens and 1 oxygen. So it makes H2O. So what is happening is a water molecule is removed. A water molecule is removed. So here this OH and here this H is removed. So what is happening? By the removal of a water molecule, a bond is formed in between these two. So what has been removed? OH is removed, H is removed. So now it has only C and O. Okay, you see this one. This one is as it is. This is this. This is this one. This is this one. These are as it is. Now here it only has C double bond O. So it has only C and O. It is joining, making a double bond. Now this one. So what happened? When the water molecule is removed from this, so what is happening? The carbon is joined to nitrogen by a bond called as this peptide bond. H2 was there. Now here to this nitrogen, 1H is there. 1 has been removed, 1 is there left over. So this is 1H. So here when these three, that is this H and these H and O has been removed in the form of a water molecule. So what is left? The water is removed and the bond is formed in between carbon and nitrogen. With the removal of water, the bond is formed between this carbon and nitrogen. So this bond, which is formed by removal of water in between this carbon of one amino acid and nitrogen of another amino acid. So this bond is called as peptide bond. This bond is called as peptide bond. Okay. So how is peptide bond formed? By the removal of water molecule. So here now these two molecules, this reaction where water molecule is removed, this reaction is called as dehydration. Dehydration. Hydra means water, D means removal, removal of water. So this reaction in this, what is happening is removal of water is taking place. So this reaction is called as dehydration reaction, or we can even call it as condensation reaction. Condensation reaction. Why? Because two amino acids are getting condensed, combined. Okay, two amino acids are getting condensed by the removal of water molecule by the peptide bond. So this reaction is also called as condensation or you can even call it as dehydration. That is, by removal of water molecule, a bond is formed in between carbon of one amino, that is in between the carbon of carboxylic group of one amino acid with the nitrogen of amino group of another amino acid. So, this bond is called as peptide bonds. So, how are proteins formed? By the peptide bond. So, this is one amino acid, one peptide bond, like this another amino acid. So, here if I take another amino acid, see here C NH2, this is H, this is R, COOH. So, what happens? Here also this one OH and this H, I will write like this NH2. So, here also one OH and one H will be removed. So, here it will be there. So, because of this removal, a bond is formed. I will remove this one. Because of this removal, so H was there, so H comes here and a bond is formed, it means between this, O goes here. A bond is formed between carbon and nitrogen. So, like this, many peptide bonds join together, many amino acids join together by many peptide bonds and form a bigger molecule called as, or when they form a bigger polymer, that is called as protein. So, that is why proteins are called as polypeptides. Why? Because they are joined together by peptide chain. So, this is one peptide, this is another peptide, this is another peptide. So, many peptides are there na. So, that is why it is called as polypeptide. Proteins are also called as polypeptides because many peptide bonds are there. Many means polypeptide bonds. So, that is why proteins are also called as polypeptides. So, this is how the proteins, that is amino acids, are joined together and form protein. By which bond? Peptide bond. It is also called as amide bond also. Amide bond also, peptide bond or amide bond. Now, coming to this one, we said that polysaccharides, they are nothing but polymers of sugars. Small sugar units are joining together and forming a bigger unit, that is polysaccharide. So, how amino acids are joining together and forming proteins? In the similar manner, the sugars, simple sugars are combining and forming a large polymer, which is nothing but polysaccharide. So, how are these sugars combining? Because sugars, one sugar is combining with another, it is combining with another. Here amino acids are combining. In this polysaccharide, sugars are combining because the monomers of polysaccharides are sugars. Sugars are combining. So, this is this glucose. This is the structure of glucose. This is the ring structure. Both of them, this is also glucose, this is also glucose, but this is the ring structure and this is two structures of glucose. So, what happens in this? If I said one glucose is combining with another glucose, how one glucose is combining with another glucose? So, here C6H12O6, C6H12O6 is the formula for glucose. So, glucose is combining and forming, like this, glucose molecules are combining and forming a big molecule called as polysaccharide. It is forming a big molecule called as polysaccharide. So, here also, in this also, when these two sugars are joining together, then they form a bond called as glycosidic bond. They form a bond called as glycosidic bond. When proteins combine, that bond is called as peptide bond. When this glucose combines, when the sugar combines, that bond is called as glycosidic. Glyco means sugar, the bond which is combining the sugar molecules. That bond is called as glycosidic bond. Glycosidic bond. Now, how it is formed? Same here also. Like here water was removed and bond was formed between this carbon of one amino acid, carbon of carboxylic acid of one amino acid with the nitrogen of amino group of another amino acid. So, bond was formed. So, here also bond will be formed. Here also bond will be formed. How they are formed? Here see this one. If this is one glucose molecule, I am not writing the whole total structure, just the end I am writing. Here this is the first part, this is the end part. So, here this is one glucose molecule, this is another glucose molecule. This is one glucose molecule, this is another glucose molecule. So, an OH from this glucose is removed and H from this glucose is removed in the form of water. In the form of water. Now, it has been removed. So, what is present? This is carbon, this is oxygen and this is carbon. So, a bond will be formed between this carbon, oxygen and this carbon. So, this bond is called as glycosidic bond. So, here I have seen we have removed the OH from here, we have removed the H from here. So, bond has been formed with the oxygen between these two bonds. So, this bond is called as glycosidic bond. In glycosidic bond also water molecule is removed. Water molecule is removed. Here also water molecule is removed. So, this is also a dehydration reaction or condensation reaction because two molecules are combining, condensing. So, this type of bond is called as glycosidic bond. If we see the structure, this is the cyclic structure of glucose. Okay. This is one glucose. I'll write another glucose. So, here will be OH. So, what happens? So, this is also OH, this is also same. So, the same structure. So, what happens? From here OH is coming out, from here H is coming out. Okay. If we see this in the structure form, so this is one glucose, this is one glucose molecule. So, what is happening here? Water molecule is removed. So, this is one H2O. I am removing this water molecule. So, what happens? What is present is oxygen. So, now here oxygen will be joining this one, these two glucose molecules. Oxygen will be joining these two glucose molecules. So, now this type of bond formation is called as glycosidic bond. This is called as glycosidic bond. Now, another polymer is nucleic acid. Another polymer which we have seen is nucleic acid. So, now in this nucleic acid, what type of bonds we see? We find ester bond as well as phosphodiester bond. Two bonds will be seen. So, how? This is one nucleotide. This is one nucleotide. In nucleotide, how many components are there? 3 components. What are there? What are they? That is nitrogenous base, phosphate and sugar. Nitrogenous base, phosphate and sugar, 3 components. So, at this first carbon of this nitrogenous base, sugar will be added. Okay? Like this, here sugar is there na, glucose is there na, glucose will be added. So, this bond is called as glycosidic bond. So, sugar is added with sugar is combined with nitrogenous base. Now when these two combine, we call it as nucleoside. How nucleoside is formed? Because of glycosidic bond. Here also one water molecule is removed and this sugar is joined with nitrogenous base. Okay. So if this is only this one, then this is called as a nucleoside. Now what is happening? Nucleotide is there. In nucleotide, one more component has came, that is phosphate group from this phosphoric acid. So this is one bond is formed here, and on this fifth carbon is attached with a phosphate group. This fifth carbon is attached with this phosphate group. So now the bond which is formed between this fifth carbon of sugar and the phosphate, this bond is called as ester bond. This bond is called as ester bond. So now we said that nucleotide is made up of three things. It is made up of phosphate plus nitrogenous base plus sugar. Okay. Now this nitrogenous base and sugar are combining with a bond. Which bond? That bond is called as glycosidic bond. Okay. And this nitrogenous base, that is fifth. So now this one, and this phosphate is attached to this carbon, fifth carbon. Phosphate is attached to this fifth carbon. Then, no, here this I said you will keep sugar in between, sugar in middle plus nitrogenous base. So the bond which is formed between sugar and nitrogenous base is the glycosidic bond. It is the glycosidic bond. Now this fifth carbon, this is the sugar, this is the nitrogenous base. If we see, this is the nitrogenous base. So here when this fifth carbon of sugar combines with phosphate, then that bond is called as ester bond. That bond is called as ester bond. Two bonds in between nitrogenous base and sugar, glycosidic bond, and in between sugar and phosphate, ester bond. Which bond? Ester bond is formed. Okay. Now this is one nucleotide. Another nucleotide is because nucleic acid is made up of many that is nucleotide plus nucleotide plus nucleotide. Many it will be joining. So instead of writing like this, I will write nucleotide plus nucleotide plus nucleotide. So like this, nucleotides will be continuously combining and forming a very big molecule that is called as nucleic acid. So now in between this one, first nucleotide and second nucleotide, a bond is formed. A bond is formed between here. This is one nucleotide. A bond is formed between phosphate of one nucleotide with the carbon of sugar of another nucleotide. A bond is formed between phosphate of one nucleotide with the carbon of sugar of another nucleotide. So that bond is called as phosphodiester bond. That bond is called as phosphodiester bond. So here this bond is called as ester bond. Okay, it is in between sugar, that is carbon of sugar, and phosphate, phosphoric acid. So that bond is called as ester bond. But here now this bond which is formed between this phosphate and the third carbon of another nucleotide. This is one nucleotide, this is another nucleotide. So if two nucleotides are joining together, how the phosphate of one nucleotide is combining with the carbon of another nucleotide. That bond is called as phosphodiester bond. So this is how the monomer units are combining together and forming a large polymer. So how proteins are formed? Protein is a polymer. How they are formed? By addition of amino acid with peptide bond. How polysaccharides are formed? By addition of monomers, that is sugars, with the glycosidic bond. And how is this nucleic acid formed? Nucleic acid is formed by 3 bonds. Okay, one bond is in between nitrogenous base and sugar, that is glycosidic bond. One bond is between carbon of sugar and phosphate. Carbon of sugar and phosphate, that is phosphorus of phosphoric acid. That bond is called as ester bond. And third bond which is formed is the bond between phosphate of one nucleotide with the carbon of another nucleotide. That bond is called as phosphodiester bond. Okay. Apart from these 3 bonds, now hydrogen bonds are also present in nucleotides. Hydrogen bonds will be, because I said you adenine and that is adenine, guanine, cytosine, thymine. These are the 4 nitrogenous bases of DNA. So adenine always combines with guanine, cytosine always combines with, adenine always combines with thymine, cytosine always combines with guanine. So these 2 combine with the hydrogen bond, formation of hydrogen bonds. 2 hydrogen bonds are joining this adenine and thymine, and 3 hydrogen bonds are joining cytosine and thymine. This we will see in detail in the next video when we see the secondary structure of nucleic acid. But as of now, we have seen how this small units are joining together to form a big polymer with the help of the different bonds. So in the next video, we will see this secondary structure of this nucleic acid.